everyone, welcome back to our channel. As usual, this is scientific lectures. If you've not yet liked and subscribed, please do so. You're missing out a lot. Yes, we still continue with our cell communication series after having looked at cell signaling, its essence, the types of receptors, the various types of cell signaling. So we go to the next method of cell communication, a method of how cells interact with each other or interact with their cytoskeleton, which is cell adhesion molecules, which I told you in our introductory video we would call CAMs. CAMs, cell adhesion molecules. We define cell adhesion molecules as proteins, normally they are proteins, located on the cell surface membrane that binds cells to cells or cells to their extracellular matrix. I said they are proteins. They are found on the cell surface membrane and they bind cells to cells or cells to the extracellular matrix. So typical of any cell adhesion molecule, it is made up of three parts, which we call domains. Number one is the intracellular domain. I'm assuming this is my cell number 1 and this is my cell number 2. So I see they're made up of three parts. The intracellular domain, which is the inside part. So this is my intracellular domain, the inside part of any cell adhesion molecule. Number 2 is the transmembrane domain, the part of the cell adhesion molecule that spans across the cell membrane. Number 3 is the extracellular domain, the part of the cell adhesion molecule that attaches it with other cell adhesion molecules or cells with their extracellular matrix. And before we proceed, cell adhesion molecules, we said they connect to cells. So that means they undergo bonding. They connect to cells. That means they undergo bonding. And there are two types of bonds exhibited by cell adhesion molecules. Number 1, it can be homophilic bonding, or it can be heterophilic bonding. When I say homophilic bonding, homo means same. So homophilic bonding is when one cell adhesion molecule binds with another cell adhesion molecule but of the same kind. Hetero means different. So it's the type of bonding where one cell adhesion molecule binds with another cell adhesion molecule but of a different kind. I hope this is clear. Homo means same. So this one, there are 4 types of cell adhesion molecules. So we have cadherins, we have what they call integrins, we have what they call immunoglobulins, super family, and we have what they call selectins. So if a cadherin binds with another cadherin, we call that homophilic bonding. But if a selectin binds with an integrin, we call that heterophilic bonding. Yes, I want us to first look at what they call cadherins. Very important to note, number 1, that we already said they undergo homophilic bonding. This means that cadherins, wherever you will find them, they will be bound to fellow cadherins. Then number 2, as we hear cadherins, that means we have calcium somewhere. So we say they are calcium dependent glycoproteins. I repeat, they are calcium dependent glycoproteins. That means in order for them to bind with other cadherins, as we had seen here, they would only do that in the presence of calcium ions. That is what I mean when I say they are calcium dependent glycoproteins. And this is the basic structure of a cadherin. And these spaces you're seeing here are what you call the calcium binding sites. These are our calcium binding sites. This is what you call the transmembrane domain. This is the transmembrane domain, the part of the cadherin that spans across the cell membrane. Then this is what you call the anchor proteins, the anchor proteins, anchor proteins, anchor proteins. For example, vinculin, and these are beta catenins. So one thing I forgot to tell you, calcium, I mean these cadherins have a specialized type of proteins called cadherins. Sorry, I was trying to mean catenins. They have specialized types of proteins called catenins. Catenins are only found with cadherins, and they will be responsible for formation of bonds. So just to recap, they undergo morphic bonding. They are calcium-dependent glycoproteins, have specialized proteins called cadherins. I mean catenins. Types, we have the types of cadherins. Types of cadherins. Number one, we have classical cadherins. These ones are summarized as pen. This is placental cadherins, epithelial cadherins, endothelial cadherins, then neurocadherins. Then you have what they call non-classical cadherins. This is why we find things like desmosomal cadherins, things like desmocolin, and desmoglin. Things like another type of non-classical cadherins, we have what they call unconventional cadherins. Things like K cadherins, R cadherins. Then lastly, we have what they call protocadherins. Things like PCHD 1, 2, 4, up to 7. Those are the different types of cadherins. Very important to know homophilic bonding, calcium-dependent glycoproteins have catenins. Then you need to know the various types of cadherins, and that is it all. Next type of cell adhesion molecule is selectins. So selectins are another form of cell adhesion molecule. Number one, they are also calcium dependent. When I say calcium dependent, I mean they only bind with other cell adhesion molecules in the presence of calcium ions. That is what I mean when I say they are calcium dependent. Number two, these ones, the difference between them and other molecules that with cadherins, they undergo heterophilic bonding. Heterophilic bonding is said binding of one cell adhesion molecule to another cell adhesion molecule, but of a different kind. Therefore, these ones can bind with immunoglobulin superfamily and integrins. These are what we are going to see next. They are calcium-dependent glycoproteins and have heterophilic bonding. They can bind with immunoglobulin superfamily and integrins. Down here is basically the structure of a selectin, and it is made up of different parts, as we saw with the cadherin. Number one, it is made up of the lectin domain. And know that whenever we have a lectin domain, that means the molecule having the lectin can bind with carbohydrates. Number two, it is made up of the EGF domain. Then it is made up of what we call the consensus repeats. Number four, it is made up of the transmembrane, the cytoplasmic tail. And this is our transmembrane domain. The transmembrane domain. The examples, I have a formula for them. I just say they are PEL. This is placental selectins, epithelial, endothelial selectins, and leukocyte selectins. Then the function, because we need to know the functions of the selectins. Number one is the responsible for cell adhesion. Number two, the responsible for leukocyte migration. Then that means they have a role in immunological responses. That is it. Lastly, they are made up of a single channel. They have a single channel. When you check in your notes, you will see something similar to that. We call that a single channel. That allows them bind with other molecules of a different kind. Next are the immunoglobulin superfamily, or they summarize as IgA. Immunoglobulins superfamily. They are called immunoglobulins because they have what we call repeated loops. You guys know a loop where you are enclosed within something. You move out, you go. When you try moving out, you go back to where you are. So these ones have repeated loops, similar to those of immunoglobulins. That is why they are called immunoglobulin superfamily. Then these ones undergo heterophilic bonding. We said heterophilic bonding is where one cell adhesion molecule binds to another cell adhesion molecule, but of a different kind. So they undergo heterophilic bonding. Number three, they are calcium independent. These are the first cell adhesion molecules that we've seen that are calcium independent, meaning that they can bind in absence of calcium ions. So basically what you're seeing here is the structure of an immunoglobulin superfamily. If I forget, they have strong disulfide bonds. What you're seeing here are sulfur bonds, and these disulfide bonds are the ones that maintain the loop-like structure. So what you're seeing here, this is the tail, then this is the immunoglobulin superfamily domain, and these are the sulfide bonds. Basically, I would say since they are similar to immunoglobulins, they are involved in their function is that they are involved in immunological responses. And we have just three types, which I usually call VIN. Vesicular cell adhesion molecules, intracellular cell adhesion molecules, neural cell adhesion molecules. That is it. The last type of cell adhesion molecules, and they are called integrins. Integrins. These guys are so special in such a way that for them they are calcium independent. They are calcium independent. That means that they don't need calcium ions in order to bind with other cell adhesion molecules. Number two, they are heterophilic. And we say being heterophilic means that they can bind with other cell adhesion molecules. For example, they can bind with the selectins, they can bind with the immunoglobulin superfamily. Then number three, they are glycodimers. And they are composed of two subunits. Number one is the alpha subunit and the beta subunit. The alpha subunit is this small one, and the beta subunit is this big one. Yes, then the other thing for them, they are so special that they don't bind cells to cells, but they bind cells to their extracellular matrix. they bind cells to their extracellular matrix. And this is just an illustration of how an integrin looks like. So what are its functions? Number one, it binds cells to their extracellular matrix. Number two, they are responsible for what we call leukocyte homing. Then number three, they are responsible for the formation of the basal membrane, which we shall discuss later in cell junctions. One thing to remember, they are calcium-independent glycoproteins. They are hydrophilic glycodimers composed of two subunits, the alpha and the beta unit. These ones bind cells to their extracellular matrix, responsible for extracellular matrix. They are responsible for binding of cells to their extracellular matrix, leukocyte homing, formation of the basal membrane. Then they are also part of the cell cycle. In fact, I had also forgotten, these ones are also part of the cell cycle. They play a certain role. Then also integrins play part in the cell cycle. And that is it for cell adhesion molecules. Thank you.